家的莅临，那也非常感谢我们的合作伙伴那个国家生技园区提供这样的场地给我们来举办这个研讨会。那也非常感谢就是我们的合作媒体就生技，他今天他大力的协助我们宣传这样子。那今天呢，我们邀请到了 Twist Bioscience， 它是一间纳斯达克有上市柜的一间生技公司。那它其实在这个美国那边其实才成立大概5年，就是上市柜才大概5年多左右。那可是其实他们在 Biopharma 的这个领域呢，其实非常的有进展。那他们其实 based on 他们的 DNA 合成技术，可是他们其实已经发展就是跟那个很多 Pharma 的那个药界其实有很深的一些联系了。那我们今天就是邀请到我们的Twist Bioscience 的 CSO 首席技术长来跟我们分享 AI machine learning 跟就是合成 DNA 文库，它是怎么去推动就是整个新药开发的进程。其实，在我们还没有办这个研讨会之前，那个办了之后，其实黄仁勋就开始在说，就是 NVIDIA 它是怎么去，他们现在也很 focus 在这个新药开发，那跟大型的语言模型，因为其实我们知道蛋白质的序列，它其实就是很多的26个氨基酸所组成的嘛。那它其实某一个方面来说，它就是一个大型的语言模型。那他们也NVIDIA 他们现在其实也投资了8家公司在就是这个生物制药相关的这个领域。那所以其实这个未来也是会是一个趋势啦。那我们今天就非常的感謝 Aaron， 他今天来到台湾来帮我们做一个这样的演讲，这样子，我们掌声欢迎一下 Aaron。
So again, I'm so happy to be here today. Again, my name is Aaron Sato. I'm the CSO of Twist Bioscience, and I also oversee a division of Twist Bioscience called Twist Biopharma Solutions. We are one of 4 divisions of Twist Bioscience, and we are focused on using all of the amazing products of Twist to actually discover and optimize antibody drugs. And so I hope through the course of my presentation today, you'll appreciate how we can use DNA to help our customers discover and optimize antibody drugs. So you might be asking yourselves, what is Twist Biopharma Solutions? So as I mentioned, we're one of 4 divisions of the company, and we're actually the merger of 2 groups that came together inside Twist. One group is a group that I started up about 6 years ago when I joined Twist, called Twist Biopharma. And it was really a kind of a genesis of a group that we started up using the core technology of Twist, which is our DNA synthesis capability, which I'll talk more about later as we get into the presentation, which was really focused on our ability to make synthetic libraries. And as you know, Twist is a synthetic powerhouse where we can use our DNA writer to make oligopools, and we use those oligopools to make high quality DNA libraries, and in particular antibody libraries, and we put them into phage display. And so today we actually have 17 different libraries all in phage display, and we use those libraries to create a whole suite of services for our customers to discover antibodies using those libraries. And so we called that Twist Biopharma, and we were quite successful at helping our customers discover antibodies from those libraries, as well as out-licensing them to our customers as well. And then about 3 years ago, we acquired a company called Veris, which was a group in Boston that was quite adept at discovering antibodies through immunization technologies out of animals. They had their own proprietary mouse. They also could discover antibodies from alpacas for heavy chain only antibodies. So it was a really nice marriage of kind of the 2 kind of core technologies of discovering antibodies in the field. In vivo-based immunization, as well as kind of in vitro-based technologies. And so it really allowed us to really provide the whole suite of services to our customers, both in vivo-based technologies as well as in vitro-based technologies. And then at the same time, I really, in looking at the field, really appreciated that in vitro AI and machine learning was really starting to come to the forefront. I really appreciate that it's really starting to become, I call it the third kind of pillar of antibody discovery and antibody optimization. And certainly we've been putting a lot of time and effort into it. And I'll spend some time talking later in my presentation about the things that we're doing to help kind of complement our approaches in vivo and in vitro discovery to help us kind of get more value from our data as we're moving through our workflows in that space. And so because of that, we rebranded ourselves as Twist Biopharma Solutions. And we actually again have a team in South San Francisco that does our in vitro and AI machine learning workflows. And we have a team in Boston that does all our in vivo workflows. Okay, so when a customer comes to us with their difficult to drug target, it really starts with antigen. So oftentimes they do have their target and their antigen in hand, but if they don't, we can help them make that antigen. We have a lot of great protein scientists in our team that can help them make their protein. And then once we have that high quality antigen, we can port that into one of two workflows, either an in vivo workflow where we can immunize animals, either again mice or alpacas, or we can port that into an in vitro workflow using phage display. As I mentioned, we have a lot of really great tools using AI and machine learning to help us maximize and get greater value from our data. And then, unlike a lot of other of our competitors, we sit inside of a DNA juggernaut, so that allows us to really drive the make test cycle. So we're not limited on terms of the amount of DNA sequences that we can make and test. So any kind of antibody sequences that come out of our workflows, we can synthesize those sequences, we can make those antibodies, and then we can do all the downstream testing. So that really kind of sets us apart from a lot of our competitors. And then finally, anytime we get leads and we want to, for example, humanize them or optimize them, again, since we're Twist and we have this DNA synthesis capacity, we can synthesize and do humanization. We can make libraries to optimize sequences. We can use phage display to help us with optimizing those sequences to make them better and optimize their specificity. In terms of our experience in the industry, between the historic Averis team in Boston and the Twist Biopharma team in South San Francisco, we've worked with hundreds of partners throughout the years, both here in Asia as well as in Europe and America. We actually have several examples of antibodies that have gone into the clinic, both from our in vivo antibody discovery efforts, as well as we actually have one example now of an antibody that's derived from our in vitro libraries that actually has gone into people just this year, which again, we're really proud of. And again, we also have examples of working with companies, not just large pharma companies, but also really small companies. So it really spans the gamut. So we really love and appreciate working with small up-and-coming biotechs because we really feel like they really see us as a partner and a really tight collaborator. But also, again, large pharma companies because we're seen as a kind of a cog in their overall machine working towards the clinic. So it really, we like kind of span the gamut in terms of all the different partners out there. And so over the years, again, we've worked with hundreds of different partners that allowed us to really play a big role in drug discovery all the way to the clinic. And in terms of the different diseases we worked in, of course, we worked in cancer, but we worked in other kinds of indications as well. Even in indications such as like animal health, for example, we actually have a really long-standing collaboration with Invetx, which is a biologics company that's actually focused on developing antibody therapeutics for animals. So that's actually a really cool collaboration we have with a US company where we're actually developing antibodies to help pets. And then in terms of different types of modalities we're working on, we're working on full-length IgGs for our customers, but also antibodies for antibody drug conjugates, heavy chain only antibodies for bispecifics, for example, and all kinds of different modalities for different engineered proteins as well. So you might be asking yourself, Aaron, there's a lot of other companies out there that do exactly what you just said. What kind of separates you from all those other competitors? And again, I'd really love to cite these kind of three core areas that kind of separate us from the pack. First one is really antibody diversity. I kind of already cited that one thing that really kind of separates us from a lot of our competitors out there is the kind of the differences in the sources of antibodies that we can actually discover, in the sense that, of course, we can discover antibodies for now in animals, in vitro libraries, and also now, I'll show you later, we can really get even more sequences from our in vitro and in vivo discovery efforts using AI and machine learning, and I'll show you how we'll do that later in my presentation. As I also indicated, because we're a DNA synthesis company, we can also go really deep into our data and really make and test really deeply. And so that allows us to really maximize the value for our customers, to allow them to get and really find those rare hits from the screening efforts that we do. And then finally, we really pride ourselves on being not only great, having great technology and be able to make and test a lot of things, but also being great antibody engineers. That allows us to really, not only once we have hits, but also be working with you to engineer your antibodies to be, again, optimize them for better potency and developability and greater specificity. And so again, we're there with you all along the way to enable you to create a much better therapeutic that can actually go into the clinic. So as I mentioned, the way I kind of see antibody discovery and antibody engineering is really in kind of three core pillars. The first one is in vivo immunization. I'll walk you through how Twist, what Twist does in this area in our Twist Boston facility in terms of our proprietary mouse, as well as what we do in alpacas. We have a lot of expertise in single B cell cloning, as well as our hybridoma with our mouse. And then we also have a lot of expertise with an array of different immunization strategies that we can do. And then second, as we discussed, we have a huge amount of expertise in developing in vitro libraries, which really rests in our ability to make DNA libraries, which again is our core expertise at Twist. And as you'll see, we have over 17 libraries, which range from scFv, VHH, and Fab libraries, which are again all in phage display. And anytime we get any of those sequences, again, we can always just make and test those sequences utilizing the make test cycle of Twist. And then we'll finally get into the third pillar, which again is something I'm really excited about, which is our ability to use AI and machine learning to initially, we really got into this to help us with lead picking. As you'll see from when we start doing a lot of our in vitro panning, particularly our cell-based panning workflows, we're doing a lot of NGS analysis of our phage display panning. And as we do that, we needed to come up with ways to actually find our leads from that NGS data. And so as part of that, we started using a lot of AI machine learning unsupervised workflows to help us find those hits. And so that's where we kind of started to dive into AI machine learning, and then we started to expand upon that. And so initially we got into lead picking, but as you'll see later, we've kind of dived into other areas as well. Okay, so kind of moving into kind of the end-to-end services that we offer. As I mentioned, it really starts with the target and the generating the repertoire of sequences. As we discussed, we can really start with, of course, the different repertoires of sequences from the animals, which again could be either mice or alpacas. We are getting into other animals as well, like rabbits. Of course, we can generate repertoires from synthetic libraries as well. We love to use, of course, different sources of generating those repertoires, which again could come from in vivo sources or in vitro libraries, or again complementing that with in silico sources in terms of then finally coming up with finding those leads and in terms of screening them. Again, we can utilize the DNA synthesis capacity of Twist to make all the DNAs of those sequences, make the antibodies, and then go through all the downstream testing to actually characterize those hits. And then if we need to, we can always optimize them for greater potency and humanize them if they're actually derived from mice. And at the end of the day, our goal is really to create development-ready candidates that our customers can then take into the clinic and actually impact patients. Okay, so let's dive into those three pillars that I talked about before, and we'll start with in vivo. What are kind of the key competencies of our in vivo offering that our customers really appreciate. And again, this is the group that's in Boston, which was the Averis team that we acquired 3 years ago. Kind of the crown jewel of this Twist Boston group is really the hyperimmune Averis diversimab mouse, which I'll talk about in a second, which has a broken immune tolerance. I'm really proud of this mouse because, as you'll see in a second, it has a broken immune tolerance that allows us to find antibodies to highly homologous epitopes. We also can discover antibodies in alpacas for heavy chain only antibodies. And again, we're also exploring other species as well. The team also has a lot of experience using single B cell cloning on the beacon. Shown on the right is just a picture of one of our scientists working on the beacon to actually discover antibodies using single B cell cloning on the instrument. And I'll show you the power of this instrument to actually screen tens of thousands of single B cells on the instrument. And then utilizing this instrument and our experience with doing really a whole suite of different types of immunization strategies, we've been able to really tackle some really tough targets like integral membrane proteins, GPCR targets, as well as a whole host of other really tough targets. So as I mentioned, I'm really proud of our mouse, which is called our diversimab mouse. It's actually it generates wild-type mouse antibodies, and it's actually on 2 different backgrounds, BALB/c and BLAC6. It's a genetically engineered mouse. It's actually on 2 different it has a broken immune tolerance. And basically what we can use this mouse for is actually what we can do is shown on the bottom left. You can actually see as we immunize this animal with targets with higher levels of homology. So you can see actually 4 different targets from 75%, 87%, all the way up to 100% homology. We can still see titer in the diversimab mouse, whereas if you look at the BALB/c mouse, you start to see loss in titer. So it really shows you as you go to higher levels of homology, tolerance starts to kick in. You start to lose binding to antibodies in this animal. Just 1 second. Sorry, it's getting a little hot in here. Yeah, sorry. 1 second. Okay, so we'll get right back to it. So again, on the bottom left, you can see again we're going through 4 different targets, and again you can really see that as we go to higher and higher levels of homology, we start to see that we can still see great titer in the diversimab mouse as we go to even 100% homology, really indicating that we can still bind to those highly homologous epitopes in the diversimab mouse. But in the BALB/c mouse, we again completely lose titer in the 100% homologous target. So it really shows you that we don't actually need to go to a knockout mouse in the diversimab setting. So we can actually access those epitopes that are highly homologous between mouse and human. Whereas if we were just using wild-type mice, we couldn't really access those epitopes because of the rules of tolerance. And then on the bottom right, you can actually see that if we look at a humanized mouse, which is still bound by the rules of tolerance, we can actually see that we still can't access those highly homologous mouse-human epitopes either. And we also can see that the diversimab mouse also can actually access a lot more epitopes in total relative to the humanized mouse as well. So it really shows you the power of this mouse relative to wild-type mice and humanized mice. We can actually find that we get a lot more antibodies in terms of sequences, as well as we can access epitopes that are oftentimes excluded in wild-type and humanized mice. In terms of the immunization strategies that the team can do in our Boston facility, it really spans the gamut in terms of the types of antigens we can use. Of course, we can use proteins, but I'm really impressed by what the team can do in terms of using other types of antigens as well. Shown on the left, you can actually see that the team can actually use DNA encoding the antigen. Oftentimes, we have examples of proteins that are actually really difficult to express, like GPCR targets. And so the team can actually immunize with DNA encoding these integral membrane proteins, where the animal actually expresses the antigen in situ, and then you can actually generate an immune response inside the animal to that target. Shown in the middle is an example where we can actually immunize with cells that overexpress that antigen. And in particular, we can do what's called a subtractive-based immunization, where we can tolerize the animal to the background cell line that's first, and then we can actually do a positive immunization to cells that overexpress that cell line. And then finally on the right is again an example of where we can again use 100% homologous protein and really show that again we can get a high titer to some highly homologous epitopes between mouse and human. So again, in the mouse setting, we can really go one of two ways. So once you have high titer in the mouse, you can really go either the hybridoma route or the single B-cell cloning route. Again, the advantage of going through hybridoma is that, again, even though it's a little slower, oftentimes, it's a great path to getting to reagent antibodies. And again, it's a tried and true method that's worked, over the years in many settings. But as I'll show you in a second, going through single B-cell cloning in the beacon is really a function-forward screen that gives you a lot of data and allows you to really kind of pick and choose the B cells that allow you to kind of find those rare B cells from your immunization. When we do our immunizations in our Twist Boston facility, we actually do a rapid immunization in our mice, so we can actually get to tissue harvest within 1 month before we then do our other fusions in hybridoma or through our single B-cell cloning on the beacon. So within a matter of a month, we can then go through the hybridoma path or the single B-cell cloning path. And now I'll walk you through the workflow for either beacon or hybridoma in the subsequent slides. And then after we go through those two workflows, we can then go through sequence recovery, where we isolate the sequence for those antibodies, and then we can synthesize the resulting sequences, make and then test those resulting sequences. So what's the advantage of working on the beacon? As I kind of alluded to, it allows us to screen tens of thousands of B cells in a very short period of time. As I'll show you in the next couple slides, we can do up to 4 sequential assays on every single B cell on the chip. And so for every single B cell, we can get a lot of data up front as we're going through the screen. So that allows us to really interrogate every B cell within that one single day as we're doing the screen. And so in a very short period of time, we can kind of home in on those key B cells from our immunization. And then, as I mentioned, hybridoma is really kind of the tried and true method for doing antibody discovery from particularly mice. And again, a lot of our customers love to use it for generating reagent antibodies, and so we continue to do that as a service for our customers. So what do we do in that single day on the beacon? If you're not familiar with how the beacon Berkeley Light System works, basically you take your B cells, and in particular what we do with our mouse B cells, which we differentiate into plasma cells, or if we're taking B cells from an immunized alpaca, we typically isolate memory B cells. We then put them on the chip, and the way the beacon works is you can use light to actually move them into these little chambers or pens. And what you do is then once each single B cell is in its own little chamber, it's actually expressing antibody in the headspace above that chamber, and then you can actually flow reagents over the headspace above each of those chambers. And so you're actually flowing fluorescent proteins above those chambers and actually trying to detect fluorescent binding events above each of those individual chambers, which indicates a positive binding event. And so in particular, you might say, for example, flow your fluorescent target over each of those different chambers with say a bead that captures your antibody. And so that can actually be used to actually detect positive binding of your fluorescent target to your antibody. And so once you can, and as I'll show you, you can actually do up to 4 different assays in that single day. And once you collect all that data, you can then export all the B cells that have all the data points that you like, and then you can PCR up all the heavy and light chain genes for all those positive B cells, and then we can sequence those heavy and light chain genes. So what kinds of assays can we run on the instrument? Again, we can run cell binding experiments, we can run ligand blocking assays, we can run multiplex, multicolor assays for example, human mouse cyno-cross reactivity. We can run functional assays if we have an assay that has a fluorescent readout. So you can really run all kinds of different really sophisticated assays on the instrument. And also in terms of that cell binding experiment, we even can run not only suspension cells, but we also could potentially even run adherent cells on the instrument. And just as an example of kind of like a case study in terms of the types of assays that we could run, this is actually an example of some customer data that we ran where on assay 1, we ran a protein binding ligand blocking assay as well as an IgG secretion assay. And from that, we actually had 274 positive hits from assay 1. In assay 2, we did a target expressing cell binding experiment. Assay 3, we ran a human cyno-murine cross-reactivity assay. And then assay 4, we actually looked to see whether the murine antibody blocked a benchmark did a benchmark binning antibody study. So from all those, 47 of them actually met all the criteria that the customer was actually focused on, and that they had human cyno cross-reactivity bound to cells and actually were ligand blockers of the 274 hits from assay 1. Okay, so now I'll jump into the second pillar of the 3, which is really our in vitro offering. And it really, again, stems from our ability to write oligopools in our oligopool writer. As you may be familiar with, this is our core competency of Twist, and it really rests in our ability to make up to 1 million individual oligos, up to 300 base pairs in length on this silicon chip. And as you might be able to see here, we can actually make oligos in these individual clusters, and within each cluster, we can actually make over 100 individual oligos. And the really cool thing about this writer is that we can actually print each individual oligo in these really tiny spots. And unlike a microarray where the oligos are actually fixed to the chip and don't come off, we can actually elute off the oligos after we synthesize them. And so when you make a gene, you actually make all the oligos that constitute that gene inside that one cluster, and then we elute off that cluster and then PCR up all the oligos together to make that gene fragment. And so that's actually the core product of Twist for our SynBio offering. We have a custom e-commerce platform where you can go online, order gene fragments or clonal genes. Again, that's our core, one of our core divisions of Twist, which is one of the other 4 divisions of the company. And again, and I'll walk you through later how we can also use oligopools for generating libraries, which is again what we used for our Twist Biopharma division to create our phage display libraries, which I'll talk about in a second. In terms of kind of some of the QC steps for the oligos that do come off our writers, where we do a lot of work to really optimize the chemistry for those oligos, because if we can really make that chemistry much more precise and much more accurate, we can make longer and longer oligos and make that chemistry much more robust. And we can make much longer and as we can make longer and longer oligos, we can potentially make longer and longer gene fragments as well. So shown here is just some examples of the uniformity of the synthesis, as well as the error rate that we have for the oligos that we synthesize. Shown here is just an example where where our error rate was 1 in 2,000. I think actually our error rate has actually gotten even better since this slide came out. I think we're actually almost up to 1 in 3,000. So it really shows you that as we've gotten our chemistry better and better and better, our error rate's actually improved over time. One thing we're also really focused on is, as you might imagine, as we miniaturize the process of DNA synthesis and miniaturize the process of making oligos, we're saving a lot in terms of the waste in chemicals. Like, we're not wasting as much in terms of all the chemicals that we're using to make all these oligos. So in terms of what a lot of our competitors use to, in terms of the chemicals they use to make oligos, we've done a lot of calculations to show that when we make oligos to make a gene, we use a lot less in terms of the overall amounts of kind of carbon footprint for the chemicals that we use relative to what our competitors use. And just as a frame of reference, manufacturing one gene is equivalent to burning for Twist, 0.04 pounds of coal, whereas a standard 96-well approach would be burning 25.8 pounds of coal. So again, it kind of gives you a frame of reference, kind of the standard approach when using a 96-well plate versus the approach that I just showed using the silicon chip technology. Okay, so again, our core SynBio offering of Twist is really our ability to make gene fragments up to 1.8 kb, or clonal genes, which again is taking a gene fragment and cloning it into your onboarded vector. And again, you can onboard any vector that you wish, and then clone in your gene fragment. If you do order clonal genes, we then sequence your clone gene, so it's a clonally perfect gene. So we not only sequence the insert, we actually sequence the entirety of the gene plus your actual vector as well. And we also have a new service today, actually called Express Genes. So back in the day, we always had scale. We always were one of the cheapest out there in terms of price, but actually today we actually are one of the fastest as well, because we've launched our Express Gene service, where we can actually deliver genes in almost less than a week now. So it's really quite amazing that we've done so much work to help optimize our overall process that Twist now is one of the most competitive DNA synthesis companies out there today, because we have, again, scale, price, and speed now. So if you aren't already ordering DNA from Twist, you should definitely check us out in terms of either gene fragments or clonally perfect genes. Okay, so now we'll get into how we use the oligo pool technology for Twist Biopharma solutions. And in particular, how do we use oligo pools to generate DNA libraries? And so as an antibody engineer, I really love using oligo pools to generate libraries, because it gives me really precise control over every single amino acid that goes into my library. In the past, I was really, I could really only use like either mixtures of nucleotides or mixtures of trinucleotides to really randomize specific sequences that went into my library. And so, as I was trying to design my libraries, there was kind of this disconnect between what I wanted to go to my library and what I could put into my library. With the Twist oligo technology, I can actually precisely design and put into my library what I can actually think. And that's because I'm actually explicitly writing specific sequences that I can actually put into my library. And so that allows me to circumvent a lot of the issues that synthetic antibody libraries have had in the past, which in the past, a lot of times synthetic libraries that have either used either mixtures of nucleotides or mixtures of trinucleotides, they've had a lot of developability issues. And so because of that, by using the Twist oligo pool technology, we can really circumvent that. And as you'll see later, the way we build libraries, we can really see that the antibodies that come out of it are actually very similar to antibodies that come out of a human being. So I'm just going to jump ahead to how we typically build libraries within the Twist Biopharma Solutions team. So we typically start from a human germline framework. So shown here is an example of, say, a heavy chain domain. And let's say, for example, we're looking at a VH3-23 heavy chain germline framework. And let's say, for example, within that human heavy chain germline framework, we can go into a human antibody database, and we can actually pull in all human. We can actually pull in all human heavy chain human CDRs, CDR1s, 2s, and 3s from that database. And actually, we can then use the Oligo Pool platform I just talked about to actually explicitly synthesize sequences for CDRs 1, 2s, and 3s. And so again, unlike mixtures of nucleotides or mixtures of trinucleotides, I can explicitly synthesize those sequences. And then I can just shuffle them in the context of that germline framework. So even though I'm synthesizing those sequences, I can actually basically, again, use a fully human germline framework and then just shuffle them in the context of that human framework. So all of the sequences that come out of the library are exactly fully human. They're a fully human framework, and at the same time, they're fully human CDRs. So every single sequence that comes out of the library is fully human. And in addition, I also can remove sequences from the library as well. I can actually remove human CDRs that might have some liabilities as well. Oftentimes, even though some CDRs, human CDRs oftentimes don't have liabilities, I can also potentially remove some CDRs that might have some developability risk from my CDR oligo pool order before we actually build the library before we order those oligos that we use to build the library. So once we then generate all those libraries, the Twist Biopharma team, as I mentioned, we've actually now generated over 17 libraries, all in phage display. We have examples of fully human Fab, SCFV, as well as VHH libraries. And as I'll show you in a second, we can do a whole series of different pannings on different types of targets. We love to use protein-based pannings, but we also can do cell-based pannings as well as peptide-based pannings. And as I alluded to in the past, when we started doing our cell-based pannings, we had to come up with a new way for us to do lead picking, and so that's why we had to come up with our AI machine learning workflows to help enable us to pick out our hits from that workflow. And so as you'll see later, by doing so, we were able to use unsupervised machine learning to use enrichment analysis and clustering analysis to help us pick out those hits. So when we take those libraries, we typically pool them based on scaffold type. As I mentioned, we have examples of Fab, SCFV, and VHH libraries. Later on in the presentation, I'll give you some examples of specific libraries that we've made. Once we take those libraries, we can do, as I mentioned, either protein or cell-based pannings. We typically do about 4 or 5 rounds of panning using phage display. If we're doing a protein panning, we can do phage ELISAs to pick out our hits, and then do Sanger sequencing to actually determine the sequences of those hits. And then, as I just indicated, if we're doing cell-based panning, we'll do NGS analysis of the later rounds of panning, and then we'll use our AI machine learning workflow that I'll talk about in a second to pick out our leads. And then once we have all those sequences, we'll then reformat them directly to our final modality, which is either IgG or VHH-Fc. We'll synthesize those hits, and then we'll make and test those IgGs for binding either in Cartera, for in SPR, or in cell binding. Okay, so one example of a library that I'm really proud of is our hyperimmune library. And we initially started with an SCFV library, but we also made a Fab library of this. And again, the dogma for antibody libraries is that heavy chain CDR3 is the most important part of the library. And so what we wanted to do is really focus in on creating a lot of diversity in heavy chain CDR3. And so what we did is we went into those human antibody databases, and we actually collated over 2.5 million human heavy chain CDR3s from those databases. And so we then took over a writer for a couple weeks, and we actually synthesized an oligo pool of over 2.5 million heavy chain CDR3s. And we then created the libraries shown here. We also created oligo pools for the other 5 CDRs of the library as well. But the bulk of the diversity was in human heavy chain CDR3. So you can see that, and then the framework that we used was VH3-23 and VKappa1-39. And then as I mentioned, we kept it in either a Fab format, shown here on the right, or we also made an SCFV format of the same diversity. And then the overall practical diversity library was on the order of 10 to the 10th. Here's just a little bit more information on the specifications of the library. Again, we had a huge amount of diversity in heavy chain CDR3. It's built off of that VH3-23 framework. And again, we have over 10 billion different antibodies in the library. And just as an example of the use of the library, we actually panned and screened it against a type 1 transmembrane protein, and actually came up with over a whole panel of sub-nanomolar binders to this type 1 transmembrane protein, really showing the power of the hyperimmune library to actually find really tight binders to this type 1 transmembrane protein. As an extension of, and again, another type of library that's really popular in our suite of libraries in the in vitro offering that we have are our VHH libraries. Again, similar to a lot of our other in vitro libraries, it really starts with a design set that we like to start with. And so of our VHH libraries, we actually started with 3,000 VHHs derived from llamas. And then we use those 3,000 VHHs to actually design the 4 libraries that I'll talk about now. The first one is actually we call our VHH ratio library. We took those 3,000 VHHs derived from llamas, which again, those 3,000 VHHs actually bound 3,000 different individual targets. And we took the CDRs from those 3,000 VHHs, and then we actually parsed them and actually pulled out the individual ratios of amino acids in each of those different CDRs, and made oligo pools that mimicked the compositions of those amino acids in CDR 1, 2, and 3. We then made a framework that was a consensus sequence of those frameworks. And then we made oligo pools again that shuffled that amino acid distribution. And we called that library our VHH ratio library. We also allowed for length variation in CDR3. And so again, this library had a lot of diversity because we again allowed for a lot of different ratios of amino acids in both CDR 1, 2, and 3. The second library is what we called our VHH shuffle library. Okay, so unlike the first library, we're allowing for a lot of ratios of amino acids, which again was kind of a departure from how we normally make libraries from what I talked about before. Here we actually just shuffle the naturally occurring CDRs, again, similar to the approach that we often take with making human libraries. Here again, we just took the naturally occurring llama CDRs from those 3,000 VHHs and just shuffled them in different combinations in the context of that consensus llama framework. The really cool thing about this library is that the final diversity of the library is actually greater than the theoretical diversity. So we're actually able to see all the combinations of CDR 1, 2s, and 3s in the library. Third library that we created is just like the last one, but we actually put it on a humanized framework. So the really cool thing about this library is that all the leads coming out of it potentially might have lower immunogenicity relative to the consensus llama framework. So a lot of our customers love to use this library because they can potentially directly use it for a human therapeutic. And then finally, the last one is our VHH H-shuffle HI library. Here, it's just like the third library, but now we're actually using that very large CDR3 oligo pool from that last library I just talked about and actually substituting it into the CDR3 position of the library. So really taking advantage of those multiple oligo pools from different libraries and actually repurposing them to create a whole suite of different libraries, and really showing you here in this context, showing you how we can repurpose a really large HCDR3 oligo pool in a smaller VHH modality. And this is really our most human VHH library because, again, it has a humanized framework as well as a human CDR3 loop. And utilizing these last two libraries, we actually did a panning using an integral membrane protein, an ion channel called P2X7, in collaboration with one of the companies that we work with in Switzerland. Here, they actually gave us some solubilized ion channel, and we actually used it for panning and actually came up with a whole suite of sub-nanomolar binders to P2X7 that also bound to P2X7 on cells, really showing the power of, again, if we have high-quality antigen, we can find high-affinity binders to, even again, a really tough-to-drug ion channel like this. Okay, now in the remaining time, I'll dive into our work that we're doing on AI and machine learning. So one way I already kind of hinted that the core use of how we use AI and machine learning is actually in lead picking. And we use it primarily to look at our NGS data for our phage display workflows. So as we're doing NGS analysis of our later rounds of panning, we're typically using our unsupervised workflows to pick out specific sequences from those phage display pannings. We're also oftentimes developing deep learning models to also find rare clones from our data, which is also, even though it does take a little longer, it's been really helpful for us to find really high affinity functional hits from our NGS data as well. Another cool thing that we've been doing is using AI and machine learning to help us with library design. We can also use a lot of our models to help us generate new antibody sequences from models of antibody databases that actually can generate new fully human antibody sequences that may not actually exist in those databases. So with knowledge of how the immune system can actually generate fully human antibody sequences, we can actually generate new antibody sequences that we can then use to actually generate new libraries of antibodies. And so we've actually done this a couple times with some external providers as well as internally at Twist to actually create libraries based off of AI and silico generated sequences. We're also doing a lot of work to use AI for lead optimization. We have a whole pipeline for doing rapid humanization, so we can actually take in sequences derived from, of course, our diversimab mouse to rapidly humanize sequences to go from mouse to human. We also are doing a lot of work to do rapid affinity maturation, both through library-based approaches, but also we're doing a lot of work on zero-shot language models, which you may be familiar with, which is this way of basically taking an initial input sequence, let's say a protein or an antibody, inputting it into a language model, and the language model basically suggests specific variants, either single amino acid or double amino acid changes to your sequence based off of knowledge of how either proteins or antibodies mutate in nature. And so we're trying to refine these models further. There's publicly available models out there already, and we're trying to either generate our own models, but also potentially refine some of those publicly available models. And so hopefully what we plan to do in the near future is potentially maybe launch some of those refined models out there and potentially use this as a service to allow our customers to rapidly use some of our zero-shot models to rapidly affinity mature sequences without actually having to go through a library build. And then finally, the last area that we're working on is creating developability filters. Can we actually take in an initial antibody or amino acid sequence and actually predict developability data? This is something that we've actually accumulated a lot of developability data over the years, and we're trying to see if we can actually establish some nice correlations between amino acid sequence and that experimental data. And we're actually starting to see some nice correlations with that experimental data. Shown here is just some of that data from that lead picking analysis that I showed before. On the bottom left is that enrichment analysis that we often do, where it's showing graphically how we look at clonotypes as we look at between abundance between rounds. Let's say in this case we're looking at rounds 2 versus 3. Shown in the diagonal is again clonotypes. If it wasn't enriched, you would expect a specific sequence to actually exist on the diagonal. if a sequence does get enriched, you would actually expect it to actually go up above the diagonal, and you can actually see in many cases a lot of sequences actually do go up above the diagonal, indicating they are being enriched as they're progressing through the rounds. And then on the right is an example of our unsupervised clustering analysis. Again, we're looking at specific features of sequences and then clustering them based on those features. So Jay often asks me, like, what is a feature? A feature could be something that's so simple as looking at sequences based off of charge or hydrophobicity, and kind of breaking them down based off different characteristics of those sequences. And so it's basically looking at breaking them down based off of their properties rather than their specific amino acid content. And then I already kind of went through this before, but we're really excited about how we can use zero-shot and few-shot models to help us optimize our sequence faster without having to go through library build. I talked about how we're using zero-shot models to just input a specific antibody sequence and give us variants. And then we've already shown that our initial models are actually giving us modest improvements in affinity. And our goal is to see if we can actually get to potentially 10-fold improvement in affinity with a zero-shot model. We're actually getting pretty close. Shown here is this data is showing that we're getting 3 or 4-fold improvement. We've actually recently almost gotten to 10-fold improvement from our zero-shot models. And then a few-shot model is actually where you take in your initial sequence and then actually layer on additional variant data that you already have. And that actually is much more powerful because you can actually use additional data that you already have to help you kind of guide the model even further. And in those cases, as you can see, we've gotten much more impactful in terms of coming up with even more additional variants to kind of focus your language model. And so in those cases, we've been able to see double-digit improvements in overall affinity quite easily. Okay, so in terms of humanization and optimization, as I mentioned, we're quite good at doing humanization of sequences. We have a really rapid pipeline for taking an initial murine sequence or initial alpaca sequence and making it humanized. And that really rests on our ability to our internal human antibody database. And then we have a really rapid AI pipeline internally where we can then look at that sequence, see how it's similar to the human antibody database, and then basically look at the junctions between the framework and the CDR regions and actually suggest a whole series of germline grafts and then make and test. And then on the right, we also can then, once we do have our humanized candidates, we also can come up with ways to generate libraries to help us optimize those candidates further. We have a system that we call TAO, which stands for Twist Antibody Optimization, where we can actually take in your initial antibody sequence and we can actually look at your antibody, and in particular the CDR regions of your antibody, and we can compare those CDR regions to the human antibody database I just talked about. And we basically look for similar CDRs in the database with a defined mutational distance that we can set. So let's say we give it a distance of 3. We look for all CDRs that are similar within 3 amino acids. And then we synthesize all of those CDRs using our writer, and then we basically shuffle all those CDRs in the context of a library. And so that basically allows us to combine our library to all and kind of explore CDR space, but again, exploring it in a natural way using our Oligo pool technology. Shown here is just kind of how we, again, go after humanization. It's just really, again, where we can, again, put our murine antibody into our AI pipeline. They suggest a whole series of graphs. We make all those DNA constructs. We do a big grid, either a 4x4, 5x5, or 6x6 matrix. We make all those antibodies and put it through a very thorough testing paradigm. And then shown on the kind of bottom right of the slide, we actually did 3 different examples of 3 different antibodies that are currently in the clinic as humanized antibodies. We actually went all the way back to the murine antibody. We rehumanized it using our pipeline, compared that back to the humanized antibody that's currently in the clinic. And in all cases, we were able to create a humanized candidate that rivaled the candidate that's currently in the clinic versus the chimeric parental antibody. We've also been able to do rapid humanization of VHH antibodies as well. This is actually another paper that we published earlier last year, where we actually, in addition to showing how we can discover antibodies out of alpacas, we also showed that we can rapidly do humanization directly out of alpacas once we get those sequences. So rather than just discovering the sequences and characterizing them as an alpaca sequence, we actually showed that once we got the sequences, before we even made and test them, we directly humanized them before we made and tested them. So that was a way for us to kind of skip the make and test part of the alpaca sequence itself and go directly to a humanized sequence. So it really showed us that our rapid bulk humanization pipeline was very robust, and shown here on the bottom right, showed us that we could actually, although it wasn't 100% successful, we were able to see at least 5 of the clones that were able to be rapidly humanized directly using our AI ML pipeline. In addition to, again, our AI ML pipeline I just talked about, another part of our SynBio offering, in addition to genes, which I talked about before, it is another extension of that is our high-throughput IgG offering, which is, again, a service that actually was kind of born out of our Twist Biopharma team. It was an idea that actually came about through working with Twist Biopharma, that in addition to making genes, we wanted to also make IgGs. And so we noticed that a lot of our customers were already ordering antibody genes. So we actually started up a new team where we started basically taking in antibody genes and then started actually doing transfections at either 1 mil or 8 mil scale in XB293. And now we're actually about to offer CHO transfections as well. So if you have any needs for making small amounts of antibody at either 1 or 8 mil scale, you can actually go online now with Twist and order antibodies either out of HEC and now out of CHO very soon. So if you have any needs there, please follow up with us as well. And the Biopharma team actually utilizes this service as part of our discovery projects. And then on the back end of, again, all of our discovery projects, we actually utilize all these services for testing all of your antibodies, but also as part of that high-throughput IgG production service I just talked about, we also can do all of this testing as well. So if you just want us to make antibodies and do a lot of testing, the Biopharma team that I just talked about can actually do that as well. So anytime we order antibodies, we can do a complementary testing service that kind of complement and give you data back as well, in addition to making antibody. And the types of assays that we can run, of course, are ELISA assays, SPR kinetic assays, flow cytometry assays, as well as a whole suite of developability assays. So just to summarize, as I mentioned, we have a whole suite of in vivo offerings, from hybridoma to single B cell cloning. We have a whole suite of in vitro offerings from our library of libraries. And then I didn't really get into it today, but really sitting at the interface between in vivo and in vitro, we also can do in vivo and immune libraries where we can immunize an animal and also make an in vitro library from that. And then one thing we're really excited about as well is that we're launching a new service, what we call our gold standard, which is really this concept that a customer can come to us with their target, and we can really kind of throw the kitchen sink at it, where we basically throw in all of our workflows at once and really kind of do a lot of these workflows in parallel to really maximize the chances of success against the target. And really the concept is that we can apply a guarantee to the target because we feel so confident that we're going to succeed against the target because we're actually applying so many technologies in parallel. And I'm just going to jump to maybe one slide and then show the final conclusion. But the last thing I want to leave you with is that we are also kind of dabbling in de novo antibody discovery in AI. The thing that we're really doing in collaboration with a de novo company called Macromoltech is that we know that we're not really experts in de novo antibody design, but we do realize that we are experts in library design. And so we're partnering with a de novo company called Macromoltech, where they actually, we gave them a target, C5a receptor, which is an integral membrane GPCR target. And we actually asked them to generate a whole series of in silico antibodies using their de novo algorithm. And so we gave them a specific germline framework. They used their in vivo algorithm to generate 10,000 antibody designs against C5a receptor. They identified 12 different druggable epitopes and then generated their 10,000 designs. And from that, we took those 10,000 designs and actually built a library from that. So really it allowed us to shuffle those 10,000 designs in a library. And really the idea is that de novo antibodies typically are not that high affinity, but through the shuffling event, we really thought that it might allow us to get to high affinity through the shuffling event to allow us to explore sequence diversity in the context of those 10,000 sequences. And so we basically, of those 10,000 sequences and shuffling it, the overall theoretical diversity again was huge, but then the final library diversity we created was on the order of 1 billion different antibodies. We did a series of panning against C5a receptor on cells, found a whole series of antibodies that bound with high affinity on cells, and we found a whole bunch of antibodies that bound with single-digit nanomolar EC50, which really again showed that we were able to find high affinity antibodies using this approach. And they were also very functional, which again was in line with almost antibodies that were actually very comparable to some C5a receptor antibodies that were actually in the clinic today. And we were actually even also able to even improve upon them even further to actually make them in line with those clinical antibodies. So you can actually see that using our affinity maturation approach, we're actually able to make them almost exactly comparable to avelumab, which was the clinical control antibody. And just as a control, we also went back and tried to see if we could how that approach compared to kind of more standard approaches, either using an immune library or just our standard and naïve libraries. And it really showed that our structural neural network approach, which I just went through, was really the most robust approach compared to those other more traditional approaches. So just to conclude, Twist Biopharma Solutions, we really have the breadth in terms of the different types of technologies. I walked you through our in vivo offering, our in vitro offering, and then all the AI and silico tools that we have. We have the quality in terms of not just the technologies, but also our DNA synthesis capacity in our test cycle. And then of course we have speed in terms of the miniaturization of our DNA synthesis and automation, but also again the expertise that we bring to help you rapidly discover and optimize your candidates. And again, we have really flexible terms, and really what we're really focused on is really making sure that we help you get to the clinic, and we're really focused on helping you have an impact on patients. And I really thank you for your time and attention, and I'd be happy to answer any questions.